So the major In the next section, we'll discuss the physiology of synaptic transmission. In this section, we'll talk about synaptic transmission and axonal transport. As mentioned before, there are some special challenges posed to neurons to allow them to communicate over long distances. The axon has its own specialized mechanisms to allow that signal to travel from the axon hillock all the way down to the termination point. In this section, we'll focus on the point of communication between one neuron and its target. The synapse itself comprises two different cells. There is a presynaptic axon and its swelling shown at the top here, and then there is a postsynaptic structure. This could be a dendrite if it was a neuron that was postsynaptic to this cell, or if this was part of the autonomic nervous system, which innervates all types of cells in the body. This could be a smooth muscle cell or some other cell in the body. Synapses have some specialized structures. Synapses have vesicles in the presynaptic membrane. These vesicles are full of neurotransmitters. Neurotransmitters actually carry the signal that traverses the synaptic cleft and will drive a postsynaptic response. Presynaptic membranes tend to also be full of mitochondria because of all the energy-dependent processes that occur at the level of the synapse. Neurotransmitter release is actually an example of exocytosis that was described earlier. Here we have neurotransmitter-filled vesicles that are docked at the presynaptic membrane. There are snare proteins, as described before, both associated with the vesicle as well as associated with the presynaptic membrane, and these snare proteins interact to allow this vesicle to dock at the presynaptic membrane. A trigger for release comes from the invasion of this presynaptic terminal by an action potential. As the action potential arrives at this presynaptic terminal, it will trigger the influx of calcium through a voltage-gated calcium channel. So these voltage-gated calcium channels, similar to the voltage-gated sodium and potassium channels described earlier, will open when the cell depolarizes. So when the cell depolarizes, or really when the terminal depolarizes, these channels open, calcium rushes in. Calcium will bind to a particular kind of snare protein that will trigger the fusion of the presynaptic vesicle to the presynaptic membrane. Neurotransmitter will get released, and that neurotransmitter will cause some response on the postsynaptic cell. So here are some examples of the types of responses that one can see once neurotransmitter is released. Neurotransmitter may bind, as shown in this case, to an ionotropic receptor that allows sodium to pass through it. So if the ligand, in this case a neurotransmitter, binds to this receptor, sodium channels open, sodium rushes in, and the cell, as shown in the bottom right, will depolarize. And when a cell depolarizes, since it's getting closer to threshold, that is, when an excitable cell depolarizes and gets closer to threshold, we refer to this as an excitatory postsynaptic potential, EPSP. It's excitatory because you're making the cell more likely to fire an action potential because you're bringing it closer to threshold. Another possibility is that the neurotransmitter could bind to a receptor, and activation of that receptor may cause the influx, in this case, of chloride. Chloride, by carrying a negative charge, will make the cell more negative, and you will move the voltage further away from threshold. This is referred to as an IPSP, or an inhibitory postsynaptic potential. Now, one can combine these postsynaptic potentials in various ways. Shown on the left is a pyramidal cell being recorded from, and a single axon impinging on that pyramidal cell's apical dendrite. As that presynaptic cell's action potential travels down the axon and then triggers a postsynaptic response, the EPSP triggered by that is shown on the bottom left. If instead of a single axon we had 3 axons impinging on this apical dendrite, and they simultaneously were all firing action potentials, one might observe a substantially larger action potential at the postsynaptic cell. This is referred to as spatial summation. You're summing up the postsynaptic potentials over space, and you end up with a larger signal than what you began with. Another way of achieving summation is for a single axon to fire multiple action potentials in rapid succession. When this occurs, the postsynaptic response, or the EPSP, does not have time to decay to baseline before the next response is triggered. Therefore, you get temporal summation. So temporal summation occurs when you sum the signals over time. Spatial summation occurs when you sum the signals over space. The combination of EPSPs and IPSPs can actually make it appear as if these neurons are really doing something akin to math. So shown here at the top is a single neuron. It has 3 axons forming synapses on it, synapse A, B, and C. A and B we'll call excitatory synapses. That is, when they get activated, you'll get an EPSP. C will trigger an IPSP, therefore it's an inhibitory synapse. So the tracings caused by activation of these individual synapses are shown at the bottom. So when synapse A is activated, one gets an EPSP as shown there at the bottom left. When synapse B is activated, one also gets an EPSP as shown. And each of these EPSPs is sub-threshold, meaning neither one by itself is large enough to trigger an action potential by causing the voltage to pass threshold. If now, shown with the pink number 2 at the bottom, if now you took synapse A and you stimulated it twice in a rapid succession such that the EPSP cannot return to baseline, one sees that the second stimulus now rides on the first stimulus and is now large enough to pass threshold and to trigger an action potential, and that's shown there with pink figure 2. This would be another case of temporal summation. Another way of getting an action potential is to simultaneously activate A plus B. So shown by pink letter 3, when A and B are both activated, neither one by themselves we know are large enough to trigger an action potential. But when simultaneously activated, they will spatially summate and allow you to have an action potential that's shown there as well under pink letter 3. Finally, if you were to activate synapse C, one will get a hyperpolarization, an IPSP. And so if one were to activate A plus B plus C together, one is left with a small potential which is not big enough to cause an action potential. This is interesting because when A and B were stimulated together, they were big enough to cause an action potential. But the incorporation of C subtracts away that potential, and you no longer have the ability to fire the action potential. So in this way, the neuron is essentially summing the activity across all of these synapses to come up with ultimately a decision point made at the axon hillock as to whether or not this cell is going to fire or not fire an action potential. So a couple of notes about neurotransmitters. Each cell has a particular type of neurotransmitter. So if a cell releases glutamate at one synapse, it's going to release glutamate at all of its synapses. Another important point is the crucial role of calcium for release. Without calcium, there will be no release of neurotransmitter. This will become important therapeutically, as we'll describe later. The release of neurotransmitter is quantal. That is, release of neurotransmitter is only through vesicles, and each vesicle will hold a certain number of neurotransmitter molecules. One cannot release only 2 or 3 or 4 molecules of neurotransmitter. You release either 1 packet or 2 packets, and so on and so forth. This is referred to as quantal release. And it's also important to remember that neurotransmitters by themselves are not inherently excitatory or inhibitory. It all depends on their interaction with their receptors. So the major neurotransmitter systems that we'll talk about today are acetylcholine. This was the first neurotransmitter discovered, for which we have quite a bit of knowledge. Catecholamines, which are found both in the brain and in the autonomic nervous system. Certain amino acids serve as neurotransmitters, such as glutamate, GABA, and glycine. Glycine is primarily found in the spinal cord. And other biogenic amines can serve as neurotransmitters, and the one that is of the most importance in this respect is serotonin. Acetylcholine. So acetylcholine is synthesized by the enzyme choline acetyltransferase. It takes acetyl coenzyme A and choline, and it transfers an acetate or an acetyl molecule from the acetyl coenzyme A onto choline to form acetylcholine. Shown in the next slide is a cholinergic synapse. The right side shows or illustrates the synapse. Here we have acetylcholine-filled vesicles that, with the appropriate stimulus, that is an action potential traveling down that axon, acetylcholine is released, and acetylcholine will bind to a receptor. Now, importantly, there are two major classes of cholinergic receptors. There are nicotinic receptors and muscarinic receptors. Nicotinics primarily are found in the autonomic ganglia as well as the neuromuscular junction, and importantly, these receptors are ionotropic, and we'll see more about this later on. Muscarinic receptors are found in the brain as well as in the autonomic periphery, and these are metabotropic receptors. They themselves are not ion channels, although they may ultimately cause the opening or closing of an ion channel. Once acetylcholine binds to either a nicotinic or muscarinic receptor, it will be broken down by an enzyme known as acetylcholinesterase. This enzyme is located at the postsynaptic membrane, and its role is to terminate the action of acetylcholine. It'll break down acetylcholine into acetate and choline, and these will be recycled back into the presynaptic membrane. Catecholamines. So the functional catecholamines that actually bind to postsynaptic receptors are dopamine, norepinephrine, and epinephrine, and their synthetic pathway is shown here. So tyrosine, which is an amino acid, is the first point of synthesis here. Tyrosine is converted into L-dopa, which is converted to dopamine, to norepinephrine, and epinephrine. There are some important points to remember about the synthetic pathway. First of all, tyrosine hydroxylase is the rate-limiting enzyme of the synthesis of all catecholamines. So if one wanted to have more production of these catecholamines, one could only achieve that through the modulation of the activity of tyrosine hydroxylase. The L-aromatic amino acid decarboxylase that converts L-dopa to dopamine, this enzyme is important clinically because the combination drug of levodopa-carbidopa, part of its mechanism of action is via the blockade of this particular enzyme. That is to say, carbidopa blocks this L-aromatic amino acid decarboxylase to prevent the conversion of L-dopa to dopamine in the periphery. Dopamine beta hydroxylase is an important enzyme because of its unique localization in the vesicle itself. The rest of these enzymes are all cytosolic. This particular enzyme is found within vesicles, and we'll show you an example of that in the next slide. And the final enzyme here is worth knowing about, this phenylethylamine N-methyltransferase, or PNMT. This is an enzyme which defines epinephrine secreting cells. Only epinephrine secreting cells will have this particular enzyme. So here is a catecholinergic synapse. Again, we've got three different catecholamines that are of importance: norepinephrine, dopamine, and epinephrine. Norepinephrine primarily is found within the autonomic nervous system. Dopamine is found in several regions of the brain, and epinephrine primarily is found in the adrenal medulla. So if we focus on the structure on the right, we can follow the synthetic pathway as it occurs inside the presynaptic cell. We have tyrosine converted via tyrosine hydroxylase to dopa or L-dopa. Dopa via this L-dopa decarboxylase is converted to dopamine. Dopamine, prior to being converted to norepinephrine, has to be pumped into a presynaptic cell, a presynaptic vesicle, and it's within this vesicle that dopamine is converted to norepinephrine via dopamine beta hydroxylase. Now, reserpine is a drug which was used in the past to treat both psychosis and hypertension, and that drug works by blocking the entry of dopamine into these presynaptic vesicles. These vesicles, for this particular kind of axon, which is a norepinephrine secreting axon, would be released when the appropriate stimulus arrives, which would be an action potential triggering an influx of calcium. Certain drugs such as bretylium and guanethidine can block the release of norepinephrine, and these drugs have been used in the past as both antiarrhythmics as well as antihypertensives. Postsynaptically, norepinephrine will bind to one of several types of adrenal receptors. There are 4 major classes: alpha 1, alpha 2, beta 1, and beta 2, and those will be discussed in more detail within the context of the autonomic physiology lecture. Once norepinephrine binds to its postsynaptic target, it will either be brought back into the presynaptic cell via an uptake 1 based mechanism. This mechanism is important because this is the mechanism which is blocked by drugs such as cocaine. A second kind of uptake mechanism is known as uptake 2, which will pump the norepinephrine into the postsynaptic cell. Once norepinephrine is pumped into either the pre or postsynaptic cell, it is further broken down. There's 2 enzymes that are important in the breakdown of norepinephrine. Monoamine oxidase, or MAO, is primarily a presynaptic enzyme. It's found in association with mitochondria. There are 2 major classes of monoamine oxidase, type A and type B. Type A is much more promiscuous. It breaks down norepinephrine, epinephrine, as well as dopamine, and as we'll learn later, also breaks down serotonin. Type B is targeted for dopamine, and for that reason, MAO-B inhibitors have been used in the past for Parkinson's disease. COMT is another enzyme that breaks down catecholamines. It's catechol-O-methyltransferase. It's found in many parts of both the nervous system and the non-nervous system. Recall that norepinephrine innervates many organs outside of the nervous system, and therefore COMT needs to be present to break this down in various other regions. It's found in the circulation, it's found in the liver, it's found in many other places where norepinephrine is active. Glutamate is found in many parts of the nervous system, usually at excitatory synapses. It binds to either ionotropic or metabotropic receptors. There are several receptor types out there for glutamate. It comes out of intermediary metabolism, out of several parts of the pentose phosphate pathway. Substrates are taken and converted to glutamate. Glutamate's packaged into presynaptic vesicles and then released. Once it's released and binds to its postsynaptic receptor, it can be shut down via either reuptake back into the presynaptic membrane, or it can be shuttled out of the synapse and be taken up by glia. The enzyme glutaminase will convert glutamate back into glutamine. There are multiple types of glutamate receptors. Some of the major types that are important for glutamate activity are the AMPA and the NMDA receptors. AMPAs are often called the non-NMDA receptors to distinguish them from the NMDAs. These are both ionotropic classes of receptors. That is, when they bind to glutamate, they themselves are an ion channel and will trigger the opening of that channel. In the case of AMPA channels, it's a relatively straightforward process. Glutamate will bind to its receptor. That receptor is a channel. The channel opens up. The AMPA channel is relatively nonspecific. Both sodium and potassium can flow through this channel. However, because of the greater driving force for sodium through the channel, more sodium enters the cell, the cells depolarize. The NMDA receptors are different. Glutamate does bind to the receptor and would normally open up this channel. However, at rest, magnesium ions are present and they block the entry of any other ion through this channel. It is only when these cells are depolarized that magnesium leaves its spot and allows both sodium and calcium to enter these cells. Another important feature of these cells is that glycine is required for the activation of these receptors. Glycine is present at high enough concentrations under most states such that it's not of pathological consequence to have glycine present or not present. The major important feature of these cells is the magnesium blockade at rest. This causes these receptors to have particularly unique functions in the nervous system. So the NMDA receptors need to be depolarized to get rid of the magnesium blockade. Therefore, these receptors make these cells good at responding to coincident inputs. That is to say that this cell will not respond to activation of its NMDA receptors unless it's already been activated by some other type of receptor. And for that reason, these cells are useful at detecting coincident inputs and therefore are potentially useful in associative learning. The calcium that enters the cell through these channels can have two different distinct effects. One is the calcium can be very important to trigger learning-induced plasticity or learning-induced changes in these cells. But recall that calcium can also be a potent inducer of cell death. NMDA receptors have been targets for multiple drugs, both in clinical use as well as drugs of abuse. So PCP is a notable NMDA antagonist. Namenda or memantine, a drug used to treat Alzheimer's disease, is also an NMDA antagonist. And ketamine, a drug used during anesthesia, is another NMDA antagonist. GABA. GABA is a widespread neurotransmitter mostly found at inhibitory receptors. And GABA actually is synthesized from glutamate via the enzyme glutamic acid decarboxylase, or GAD. GABA is packaged into presynaptic vesicles and then released, and its uptake is either through a non-specific reuptake system shown here in the figure, or it's shuttled into glia, and then ultimately GABA is broken down by GABA transaminase. The GABA receptor is another form of a ionotropic receptor. When GABA binds to this receptor, it will allow calcium influx into the postsynaptic cells, assuming that the voltage is appropriate for that to happen, and that will cause the postsynaptic cell to hyperpolarize. There are multiple compounds that can alter the influx of calcium into these cells. Drugs in the benzodiazepine class, such as Valium, Xanax, and multiple others, as well as barbiturates, can potentiate the influx of calcium. Their mechanisms of action are slightly different. Both of these are allosteric modulators, that is, they will only work in the presence of GABA. Benzodiazepines will increase the frequency of channel opening, whereas barbiturates will increase the duration of each open time. However, they both will potentiate the total amount of chloride that enters the postsynaptic cells caused by GABA. Serotonin also has a widespread distribution, primarily as a neuromodulator. This compound is thought to be important for mood regulation. Serotonin itself is synthesized from tryptophan, packaged into presynaptic vesicles, released, and it, unlike glutamate or GABA, has a very specific reuptake system, and this specific reuptake system has been a major target for antidepressants known as the SSRIs. These are the specific serotonin reuptake inhibitor classes of drugs. And then finally, the neuromuscular junction is another example of a cholinergic synapse. This is the synapse where we know probably the most about its function. We'll discuss this in more detail in the context of our neuromuscular physiology lectures. The important points to remember about these synapses is that acetylcholine is released, it binds to a nicotinic form of a receptor, causes depolarization of the postsynaptic cell, in this case a skeletal muscle cell, that will trigger an action potential and then muscular contraction. The rest of the details about these mechanisms with respect to muscle physiology will be discussed in the context of the neuromuscular physiology lectures. There are several diseases that involve synapses, and we'll discuss the clinical implications of alterations of synaptic physiology. So certain diseases that are specific to the synapse are as follows: myasthenia gravis, Lambert-Eaton myasthenic syndrome, botulinum toxin, black widow spider toxin, and the tetanus toxin. And we'll talk about all of these. And there are drugs that are specific for the synapse, such as the neuromuscular blockers, and again, we'll discuss these as well. Myasthenia gravis is an autoimmune destruction of postsynaptic cholinergic receptors. During this disease, the receptor count will decrease on the postsynaptic cell, and the muscle end plate, which normally has a rugated appearance, will flatten. The clinical manifestations of this are weakness, ptosis, dysphagia, and very characteristically, these patients complain of fatigable weakness. The major form of treatment of these diseases is to treat with drugs that block the acetylcholinesterase enzyme that normally breaks down acetylcholine, therefore allowing a longer duration of action of the released acetylcholine at the neuromuscular junction. The other form of therapy here for this disease obviously is to treat the underlying immune problem of the disease with immunomodulatory agents. Lambert-Eaton myasthenic syndrome is a similar disease in clinical manifestations, but quite different in terms of the locus of disease. This is a presynaptic autoimmune disease, often paraneoplastic. So rather than insufficient activation of postsynaptic receptors, the locus of the disease here being presynaptic causes insufficient release of acetylcholine. The target of the immune response here is the voltage-gated calcium channels that are crucial for the influx of calcium and therefore crucial for the release of neurotransmitter. These patients have the unique phenomenon of facilitation via pre-exercise, which works by increasing presynaptic levels of calcium. The treatment of this disease is also with cholinesterase blockers, drugs like pyridostigmine and neostigmine. These will prevent the breakdown of acetylcholine and allow a longer duration of action of the small amounts of acetylcholine released by these patients at the neuromuscular junctions. One can also use drugs that modulate the immune system, and importantly, since this disease is often paraneoplastic, a major therapeutic focus is to treat the primary malignancy if there is one. Diagnostically, one can distinguish in a quite straightforward fashion between myasthenia gravis and Lambert-Eaton myasthenic syndrome. In myasthenia gravis, if one were to apply repeated stimulation, one would find a decrement of the response. Shown on the top right, the left-hand figure of the top right overall figure shows a normal response to repetitive stimulation, and that is there's no drop-off in the response of muscular strength or muscular activation with repetitive stimulation. If one did the same test with somebody with myasthenia gravis, one would find a drop-off in the response over the course of the stimulation period. In Lambert-Eaton myasthenic syndrome, shown with the pink letter A, pre-stimulation causes a standard amount of activation of some muscular group. However, when tested again a short time later, one finds that this pre-activation causes a potentiated or a larger response, shown in figure B. Figure C shows repetitive activation of a single muscle group, and one can observe a gradual increase in muscular strength over the course of that stimulation period. Another disease of the neuromuscular junction is that induced by the botulinum toxin. The botulinum toxin will enter the alpha motor neuron at the motor end plate and will inhibit excitation secretion coupling by specifically targeting the snare proteins. The ultimate result of this will be less release of acetylcholine, therefore less muscular contraction and a flaccid paralysis. This toxin has also been used as a drug for cosmetic purposes, as shown in this slide. black widow spider toxin also targets the neuromuscular junction. However, in this case, instead of inhibiting the release of acetylcholine, one achieves a greater amount of acetylcholine release for any given presynaptic activation. However, this excess release of acetylcholine has a biphasic response. Although it can cause continuous stimulation and tetany early on, because of the inability of the postsynaptic cells, in this case skeletal muscle cells, to repolarize, they ultimately become weak, and despite initial excess contraction, one will find a flaccid paralysis at the end stage. The tetanus toxin is another toxin which affects neuromuscular activation, although not directly at the neuromuscular junction. The tetanus toxin enters through a peripheral source, and this toxin travels neural pathways and will travel retrograde and will cross synapses. Ultimately, this toxin will target GABA and glycine secreting neurons in the spinal cord and the brainstem through targeting of snare proteins in those neurons. Without the activation of inhibition, one will have excess excitation and will have the inability to control muscular contraction. And this creates some of the very typical clinical phenotypes as shown on the right, the sardonic smile and the posturing of the patient on top, and the ophthalmotonic posturing of the patient on the bottom. There are also several drugs that specifically target the neuromuscular junction. So drugs such as curare, which are derived from the natural product curare, block nicotinic receptors via competitive inhibition and will lead to a flaccid paralysis. These are used during neuromuscular blockade during anesthesia. Succinylcholine will also cause neuromuscular paralysis, but in a very different manner. These compounds will bind to nicotinic receptors. They will activate these nicotinic receptors, but after triggering an initial action potential, will not release from the receptor. The maintenance of open sodium channels will cause the inability of the membrane potential to become reestablished, causing a depolarizing blockade of these postsynaptic cells. Therefore, early on, one may see tetany with this drug, but this is followed, similar to the case of the black widow spider toxin, with a flaccid paralysis. The clinical pearl here is that too much or too little acetylcholine can disrupt neuromuscular transmission and cause weakness. This concludes our discussion of the physiology of synaptic transmission.